Hey, before we start, I want to be clear about something. What we're about to talk about is not scary because it's complicated. It's actually the opposite. Cancer begins with something simple, almost mundane. Cells doing what they normally do, growing, dividing, maintaining your body. And then something small shifts. One tiny mistake in one cell. Nothing dramatic, nothing obvious. But that one mistake can set off a chain of events that compounds over time, building silently, accumulating damage until suddenly the system that was supposed to protect you becomes the thing that threatens you. So stick with me here. This isn't about doom. It's about understanding what your body is actually doing every single day, all the time, and how an incredibly orderly system can gradually come undone. Because knowledge here matters, not because it makes you anxious, but because it helps you understand what's actually happening beneath your skin. When you understand the mechanics, when you understand how these processes work normally, you can appreciate how extraordinary it is that your body maintains such a delicate balance most of the time. You can see why even small errors, repeated and accumulated, become dangerous. You can see how a single point of failure, one broken gene, one disabled checkpoint, can eventually unravel the entire system. Understanding these mechanisms is the first step toward understanding not just what goes wrong, but why. Let's ease into this. Not rushed, just clear. Just honest about the mechanisms and the incredible systems that keep us alive. Your cells are working right now. And to understand what goes wrong in cancer, we need to start by understanding what goes right. Normal cell function forms the baseline we need to grasp before anything else makes sense. Imagine your body as a massive city. Billions of cells, each one a citizen, each one with a job. They're not thinking about it. They're not planning. They're just following instructions that have been written into their biology, instructions that have been refined over millions of years of evolution. These instructions are incredibly detailed. They tell a cell when to grow, when to divide, when to stop growing, when to prepare for reproduction, when to rest. Some cells are permanent citizens who never divide after they mature. Neurons in your brain, for instance. Once they're grown, they stay. Some of them have been in your head for decades without dividing at all. But other cells are constantly cycling through this process of growth and division. Skin cells, blood cells, cells lining your gut. These cells are born, they grow, they do their job for a limited time, and then they die and get replaced. It's constant renewal, like a city where buildings are always being constructed and demolished, but the city itself stays stable. Your gut lining replaces itself every few days. Your skin sheds millions of cells every single day. Your blood is being regenerated continuously from stem cells in your bone marrow. About 2 million red blood cells are produced every single second in your body. 2 million, just to replace the ones that are dying. This is not a static system. It's a living, breathing process of constant replacement and renewal happening every moment you're alive. And it works, which is remarkable when you think about it. The coordination required is staggering. The precision required is almost unimaginable. Billions of cells all following the same rules, all communicating through chemical signals, all working toward the goal of maintaining a functional body. This stability depends entirely on control. Cells don't just divide whenever they feel like it. That would be chaos. You'd inflate like a balloon made of cells until you exploded. You'd have a tumor the size of a person because cells somewhere decided to keep dividing indefinitely. Instead, cells have mechanisms, built-in governors, systems that say, yes, divide now, or no, wait. Systems that check if conditions are right, if energy is available, if the DNA is intact, if everything is set up properly for the next division. These aren't conscious decisions. There's no committee meeting in your cells debating what to do. They're purely biochemical processes. Proteins binding to DNA, triggering cascades of reactions, each step checking the previous step. One protein activates another, which activates another, which sends a signal to the nucleus, which reads a specific gene, which produces a product that either accelerates the process or halts it. These are physical, chemical events. Molecules fitting together like keys in locks. When the key fits, something happens. When it doesn't fit, nothing happens. The precision is extraordinary. A protein might have a specific shape that only fits with one other molecule out of thousands in the cell. It's like finding one specific puzzle piece in a pile of thousands and having it slot perfectly into place every single time. The machinery is intricate but precise. And precision matters because the consequence of imprecision is uncontrolled growth. Now, most of the time, this works perfectly. Your body maintains this incredible balance. Billions of cells all following the same fundamental rules, all communicating through chemical signals, all knowing when to stop and when to go. It's orchestrated without a conductor. It's coordinated without a central command center. Each cell is making its own decisions based on local information and global signals. But the system relies on something fundamental. It relies on the instructions being clear. It relies on the communication being accurate. It relies on cells being able to read the signals properly. It relies on the breaks being intact. And cancer begins when the reading fails. When one cell, just one, gets confused about what the instructions actually say. Or worse, when it stops listening to the instructions altogether. DNA and genetic information are the actual instruction manuals that cells follow, and understanding this is crucial because this is where things start to go wrong. Your DNA is like a recipe book, but not a simple one. Not like a recipe for bread, more like a complete manual for building and maintaining a human being, then keeping that human alive and functioning for decades. Every cell in your body contains a copy of your entire genome. 3 billion base pairs of genetic code. 3 billion instructions arranged in a precise order. 3 billion chemical letters, each one potentially carrying information. If you tried to write out your genome by hand, at a rate of 10 base pairs per second, it would take about 10 million seconds to write the whole thing. That's more than 100 days of continuous writing, 24 hours a day. That seems overwhelming, and it is. But the key thing to understand is that most of your cells only read a small portion of that manual at any given time. A liver cell reads the instructions for being a liver cell. It's reading maybe 20,000 genes out of the 20,000 total genes you have. Wait, that's more than I said. Actually, most cells read most of their genes to some degree, but they prioritize. A heart cell reads the genes that code for heart-specific proteins. It reads the genes for the proteins that make muscle fibers contract. It reads the genes for the ion channels that control electrical signals. A brain cell reads completely different genes. An immune cell reads yet another set. Same DNA, same genome, but different parts are being used. Different genes are switched on and off. Genes are specific sections of DNA that code for proteins, and proteins are what actually do the work in your cells. Proteins are the workers, the tools, the machines. They build structures, they speed up chemical reactions, they transport other molecules, they signal for changes, they do everything. DNA is the blueprint. Proteins are the construction crew. So when we talk about genetic changes, what we're really talking about is changes in those instructions, changes that might tell a cell to make a different protein, or make more of a protein, or make less of a protein. These changes happen constantly. Mistakes occur during DNA copying. Environmental factors damage DNA. UV radiation from the sun can cause DNA damage. Tobacco smoke contains chemicals that bind to DNA and cause mutations. Some mistakes are spontaneous, just errors in the cellular machinery that copies DNA. These mistakes are usually caught and repaired. Cells have quality control mechanisms that are almost paranoid about accuracy. Repair proteins patrol the DNA looking for damage constantly. They find mismatched base pairs where the wrong letter got inserted. They find breaks in the DNA strand where one or both strands are severed. They find distortions in the double helix where something doesn't look right structurally. They cut out the damaged section and patch it with the correct sequence. It's like having multiple proofreaders who are constantly checking your work and fixing errors before they become permanent. But the repair systems don't just passively wait for damage to show up. Some repair proteins actively scan the DNA, checking for errors. Some repair systems are triggered only when the cell is not actively replicating. Some are triggered by specific types of damage. There are different pathways for different kinds of problems. It's a multi-layered defense system. The precision of these repair systems is extraordinary. They can find and fix damage in a 3 billion letter document. They can distinguish between normal DNA and damaged DNA, but they're not perfect. But sometimes a mistake slips through. Sometimes the repair system doesn't find the damage. Sometimes a repair attempt fails. Sometimes it gets copied into the next cell division. Sometimes it gets into a part of the code that actually matters for cell growth. And that's when things start to get interesting. Your DNA also contains sections that are not protein coding. These were once thought of as junk, useless sequences between the real genes. Just filler, like the blank pages at the back of a notebook. Scientists called them junk DNA, which says something about how much we didn't understand. But we've learned in the last couple of decades, especially with genetic sequencing technology improving, that many of these sections are actually regulatory regions. They're incredibly important. They control when genes turn on and off. They control how much of a protein gets made. They respond to signals from other parts of the cell. They're like switches and dimmers on a light system. A gene might be fine, perfectly intact, coding for a perfectly normal protein, but if the switch that controls it gets broken, the gene might turn on all the time, or turn off when it shouldn't, or behave erratically. Maybe it turns on when it should be off. Maybe it makes 10 times more protein than it should. Maybe it makes half as much. These regulatory changes can be just as dangerous as mutations in the genes themselves. Cancer often involves these kinds of changes. It's not always that the gene itself is broken. It's that the regulation of the gene is broken. The instruction might be fine, but the signal to stop reading it got corrupted. This is important because it explains why people with the same genetic mutation might develop cancer at different rates, or in different tissues, or at different times. The DNA damage itself is just the beginning. What matters more is context. What part of the code was damaged? What other systems are in place to catch it and repair it? Whether the cell has other problems already. DNA damage is so common that cells have evolved multiple systems to repair it. Multiple backup systems, multiple layers of quality control. But these repair systems themselves can be damaged. So you can end up in a situation where a cell is accumulating errors because the system meant to catch errors is itself broken. It's a cascading failure. One mistake damages the system that catches mistakes. The next mistake goes undetected. The next one goes undetected too. The errors compound. And cancer often requires multiple cascading failures before it actually begins. Cell division basics describe how cells actually grow and prepare to split into 2 new cells. And this is where the precision becomes critical. Understanding the cell cycle is understanding the rhythm of life at the cellular level. A cell starts in what's called G1 phase, which is basically the growth phase. The cell is reading its DNA, making proteins, accumulating resources, physically getting bigger. This can last for hours, sometimes much longer, depending on the type of cell and the environmental conditions. Some cells go through this cycle very slowly. Some cells divide every 24 hours. Some cells only divide once in a lifetime. The length of G1 is not fixed. It's one of the flexible parts of the cycle. The cell needs to reach a certain size before it moves forward. It needs to have enough energy and enough building blocks, enough ribosomes to make proteins, enough mitochondria to produce ATP, enough of everything to split into two functional cells. These aren't abstract requirements. A cell that's too small cannot successfully divide into two functional cells. It's a practical constraint rooted in basic biology. You can't just keep dividing indefinitely without growing. Eventually, you'd run out of cellular material. While in G1, the cell is also checking its DNA carefully. Is everything intact? Any damage? Any mutations in critical genes? Cell decides it's ready. Once it reaches cell decides it's ready. Once it reaches the right size and the right signals arrive, it moves into S phase, which stands for synthesis. This is when DNA replication happens. The cell needs to make a complete copy of all 3 billion base pairs of its genome. This copy needs to be accurate, not 99% accurate, not 99.9% accurate, extremely accurate. When you're copying 3 billion pieces of information, even an error rate of 1 mistake per billion is too much. At that rate, you'd accumulate multiple errors every time you divide. So the cell has proofreading mechanisms built into the replication process itself. Special proteins follow right behind the copying machinery, checking each base pair, fixing mistakes as they happen. It's like having someone following behind you as you write, correcting typos instantly before they become part of the document. Despite all this built-in quality control, some mistakes still happen, but they're rare enough that the system can usually handle them. S phase typically takes between 6 and 8 hours depending on the cell. During this time, the cell's DNA is being read simultaneously by hundreds, thousands of copying machines, all working on different sections at the same time. They're called replication forks because they create a fork-like structure in the DNA as the two strands separate. They start at multiple points on the chromosome and move toward each other, copying as they go. Think about it. The cell doesn't just have one copying machine. It has thousands of them working simultaneously. The whole genome is being replicated in parallel. If all these machines make errors or interfere with each other, that's a serious problem. Replication could break down. Sections could get copied twice while others get skipped. A gene could be duplicated. A critical region could be deleted. So there's coordination. There has to be communication between the different replication complexes. Signals that say, I'm starting here, you start there, make sure we meet properly in the middle. License this region, don't license that region, making sure they don't crash into each other or leave gaps. The machinery of replication is so complex that it's hard to overstate it. There are helicases that unwind the double helix. There are single-strand binding proteins that protect the unwound strands. There are primus enzymes that lay down primers. There's DNA polymerase that adds the new nucleotides. There are ligases that seal the gaps. And there are proofreaders checking everything. It's like an assembly line factory, except the product is life itself. Once replication is complete and verified, the cell moves into G2 phase, which is another growth phase, but with a specific purpose. The cell continues to grow and accumulate resources, but now it's specifically preparing for the actual division. It's making the proteins that will be needed to pull chromosomes apart. It's making the proteins that will form the cleavage furrow that pinches the cell in half. It's organizing its cellular structures, repositioning the centrosome, preparing the spindle apparatus. If DNA damage was found and not repaired by this point, another checkpoint catches it. Another chance to stop and fix or to self-destruct if the damage is too severe. Then comes M phase, mitosis, the actual division. The DNA that was replicated is now condensed into visible structures called chromosomes. The DNA unwinds and lies loose normally throughout the cell, but before division, it gets wound tightly, coiled and supercoiled, until it's so dense it becomes visible under a regular microscope. It's like taking a filing cabinet's worth of papers and compressing them into an origami sculpture. Each chromosome has been replicated, so there are 2 copies of each one, stuck together like Siamese twins joined at a point called the centromere. Machinery pulls these copies apart to opposite ends of the cell. It's like having 2 teams pulling in opposite directions on a rope. The spindle fibers that do this pulling are made of tubulin protein, assembled and disassembled dynamically. Meanwhile, the cell is pinching itself in half at the equator, like a balloon being tied off in the middle. The membrane at the equator is drawn in by protein filaments that contract, the same filaments that create muscle contractions in your muscles. They're called actin filaments, and they're incredibly strong. They pull inward with tremendous force. When the pinch is complete, you have 2 daughter cells, each with a full copy of the genome. Each one is ready to grow and divide again if needed, or to settle into its specific role in the tissue. Each one is a clone of the parent cell, carrying the same genetic information. If the parent cell was normal and healthy, both daughters inherit normal, healthy genomes. If the parent cell carried a mutation, both daughters inherit that mutation too. But here's the thing that matters for understanding cancer. This entire process, from G1 through G2 and into M phase, is tightly regulated. It's not automatic. The cell is constantly checking, asking questions, making decisions at multiple points. And those decisions are made based on signals from outside the cell and messages embedded in the genes themselves. A growth factor arrives from neighboring cells, a signal saying, "We need more cells here." The cell receives that signal. Its internal machinery processes it. Specific proteins activate. Specific genes turn on. But the cell also checks, am I big enough? Is my DNA intact? Is this really the right time? A cell doesn't divide because it wants to. It divides because it received signals telling it to divide, and because its internal checks say that division is safe. If those signals get scrambled, if those internal checks stop working, the process doesn't pause. It accelerates. And once it starts accelerating, it becomes very hard to stop. Checkpoints and control systems are the safety mechanisms that keep cells from dividing when they shouldn't. And understanding these systems is essential because cancer is fundamentally about checkpoints failing. Your cells have learned through millions of years of evolution that uncontrolled growth is dangerous. It threatens the whole organism, so they installed brakes. They built in pauses. They created moments where the cell has to verify that everything is okay before moving forward. These checkpoints are not unique to humans. Single-celled organisms have them. Yeast cells have them. Cells in fruit flies have them. This is a deeply conserved mechanism which tells you how important it is. There are 3 major checkpoints in the cell cycle. The first one happens near the end of G1, and it's called the restriction point in some organisms, or the G1 checkpoint in others. This is the point where the cell has to commit. Once you pass this checkpoint, the cell is committed to dividing. It's a point of no return, at least for that division. Up until this point, a cell can change its mind. It can respond to new signals. It can back out. But once it passes the restriction point, the machinery is set in motion. DNA replication is going to happen. The cell is asking critical questions before making this commitment. Am I big enough? Do I have enough energy? Is my DNA okay? Do I have the right signals telling me to divide? These aren't yes or no questions. They're more nuanced. The cell is evaluating conditions, checking energy levels, measuring size relative to requirements, reading chemical signals. If the answer to any of these questions is concerning, the cell doesn't proceed. It stops. It stays in G1. It waits. It waits for conditions to improve or for it to receive the right signals. It might wait for hours or days. Some cells wait until they die, never dividing. Some cells exit G1 permanently and enter a quiescent state called G0, where they're alive but not actively cycling. They're waiting. Think of it like a security checkpoint. Papers are checked. Temperature is taken. The person either goes through or they wait, but they don't force their way through. If a cell does force its way through without permission, without those internal checks being satisfied, that's bad. That's the beginning of the problem. That's when errors start to accumulate. This checkpoint is controlled by proteins called cyclins and something called CDKs, which stands for cyclin-dependent kinases. These are molecular clocks. They appear at specific times during the cell cycle. They push the cycle forward, and then they disappear. Cyclin D and CDK4 or CDK6 are the ones that control the G1 checkpoint. They build up gradually during G1. They form complexes together. Cyclin D clasps CDK4, and this complex becomes active. This complex then goes and attaches to other proteins, activating them. They activate another protein called RB. Wait, that's actually something they inactivate. They phosphorylate RB, which is a protein that normally says no to cell division. Once RB is inactivated, it can't say no anymore. So the cell proceeds. The machinery accelerates. But there's a break on this system, a crucial break, a protein called P53. This is important. Listen to this. If DNA damage is detected, if the cell's surveillance machinery finds problems in the DNA, P53 gets activated. Once activated, P53 does something critical. It activates another protein called P21. P21 then goes and binds to the CDK4 cyclin D complex. It wraps around it. It blocks its activity. It's like slamming on the brakes. The cell stops. It cannot pass the checkpoint until the damage is repaired or until the cell kills itself through a process called apoptosis. P53 is so important that it's been called the guardian of the genome. It's mutated in more than half of all human cancers. Think about that. If you lose P53, if that gene is broken, you lose a critical line of defense. You lose the ability to stop cells that have damaged DNA from dividing. You lose the ability to tell a cell with unfixable damage to kill itself. It's like dismantling a security system that's been protecting your body for decades. The cell becomes dangerous. The second major checkpoint happens at the G2 to M transition. The cell is about to divide. It's had its DNA replicated. Now it needs to verify that the replication was accurate. It needs to verify that the chromosomes are properly condensed and ready to separate. It's the final quality check before the expensive process of mitosis, before the point of actually splitting. If there are problems at this stage, if DNA damage is detected, if the cell isn't ready, the cell stops again. It's controlled by similar systems. Cyclins and CDKs, but also P53 can activate here too. This checkpoint also looks at DNA damage. Are there breaks in the DNA? Double-strand breaks where both strands of the helix are broken. Tangled chromosomes? Problems with the machinery that will separate the chromosomes? If yes, the cell stops. It tries to repair. Or if the damage is unfixable, it dies. The third checkpoint happens during mitosis itself, during the M phase. This one is called the spindle checkpoint Because it monitors the spindle, which is the machinery that pulls chromosomes apart. The spindle is made of protein fibers called microtubules. These fibers attach to structures on the chromosomes called kinetochores. The spindle checkpoint checks whether each chromosome is properly attached. Each chromosome should be attached to spindles from both poles. If a chromosome is loose, if it's not properly connected, if the spindle fibers aren't pulling equally from both sides, the checkpoint activates. The division pauses. The cell tries to fix the attachment. Proteins readjust. The spindle realigns. If it can't be fixed, if the attachment is still wrong when the cell checks again, the cell might kill itself rather than produce a daughter cell with the wrong number of chromosomes. Why would a cell kill itself? Because a daughter cell with the wrong number of chromosomes is often more dangerous than no daughter cell at all. These checkpoints are not obstacles that cells resent. They're not rules imposed from outside. They're systems that cells have built into their own code, because cells, over millions of years, learned something crucial. They learned that making mistakes in replication is worse than being careful and slow. A cell that divides with damaged DNA creates two broken cells, two cells that carry the same damage, two cells that might divide again, spreading the damage further. Each division spreads the error to two cells, then 4, then 8. The damage exponentiates. Meanwhile, a cell that pauses, that takes time to fix the damage, that kills itself if the damage is unfixable, that cell is protecting the whole organism. It's playing the long game. It's making a sacrifice for the survival of the whole body. Cancer disrupts this game fundamentally. Cancer begins when the checkpoints fail. When a cell gets a mutation in p53, for instance, it loses its ability to stop at the G1 checkpoint when DNA is damaged. It loses its ability to activate p21, so it divides anyway, with broken DNA. That broken DNA gets copied during the next round of replication. Both daughter cells now have the same broken DNA. Plus, whatever new errors arose during their replication, if they divide again, they create 4 cells with multiple mutations. 8, 16, 32. The error compounds. Each division spreads the problem further. The system that was supposed to protect you starts working against you. It works to spread cellular errors. And the scary part, is, it often doesn't feel like a dramatic moment. There's no alarm. The cell doesn't scream. It just divides. And then it divides again. Nobody notices. The tissue doesn't feel different. Your body doesn't alert you. The immune system might not recognize that something's wrong yet. 1 bad cell becomes 2. 2 become 4. 4 become 8. After 10 doublings, you have over 1,000 cells with the same mutation. After 20 doublings, over 1 million. After 30 doublings, over 1 billion. And even then, with 1 billion cells that have a mutation, the tissue might still look and feel completely normal. It's only after many more doublings, after decades of accumulation, that things become visible, that tumors form, that cancer becomes detectable, and by that time, the cell has accumulated multiple mutations. Not just the loss of p53, maybe also a mutation that makes it produce too much of a growth-promoting protein, maybe also a mutation that prevents it from dying when it should, maybe also a mutation that helps it ignore the signals from neighboring cells telling it to stop. Each mutation adds to the problem. Each one takes away another layer of protection. Each one removes another break. And nobody notices at the tissue level until years or decades later, when the accumulation of these errors has created a cell that's fundamentally different from what it should be. A cell that stopped listening to the signals that keep the body orderly. A cell that's crossed the invisible line from normal to cancerous. That's how it happens. Not with drama, not with a bang. Quietly, cell by cell, error by error, codes for these checkpoint proteins. The codes for these checkpoint proteins, the system doesn't just malfunction. It fails, sometimes completely. The brakes don't just fail gradually. Sometimes they disappear entirely. And that's when things get dangerous in ways that are hard to overstate. The failure of control systems is where cancer actually begins. Not in the existence of damage, because damage is constant and expected, but in the failure of systems to detect that damage and respond appropriately. Let's say a cell develops a mutation in the p53 gene. Remember, p53, the guardian of the genome. This protein is supposed to activate when damage is detected. It's supposed to tell the cell to stop, to pause, to give the repair systems time to fix the problem. But if p53 itself is broken, if that gene carries a mutation that prevents it from being made properly, or that makes the protein it produces non-functional, then the entire system of checking for damage collapses. The cell with broken p53 can have severely damaged DNA and still pass right through the G1 checkpoint. The damage doesn't matter because the system that's supposed to detect it and sound the alarm doesn't work anymore. It's like having a smoke detector with dead batteries. The fire could be raging, but nobody gets the alert. The cell divides anyway, and it passes that broken p53 gene to both of its daughter cells. So now you have two cells with damaged DNA and non-functional p53. They divide again. 4 cells, 8, 16. The problem exponentiates because the system that's supposed to contain the problem doesn't work. Each division spreads the broken checkpoint to more and more cells. This is how a single mutation in a single critical gene can start an avalanche. Checkpoint failure creates a vicious cycle. A cell with broken p53 is more likely to accumulate new mutations because it's not stopping to check for and repair DNA damage. It's just dividing all the time with broken DNA. So the daughter cells don't just inherit the broken p53. They inherit whatever other mutations were present in the parent cell, plus new mutations that accumulated during the parent cell's division. The error burden keeps growing. The mutations compound. After 5 divisions, a cell that started with one p53 mutation might have accumulated multiple additional mutations scattered throughout its genome, each one potentially damaging another protective system. After 10 divisions, dozens of mutations, the original single-point mutation that broke p53 has cascaded into a widespread pattern of genetic damage, and the cell that started with this single critical failure is now a genetic mess. A cell that's accumulated enough mutations that it barely resembles a normal cell anymore. This is why cancer cells look so genetically bizarre when researchers examine them. They're not just slightly different from normal cells. They often have dozens or hundreds of mutations. Some mutations that are clearly cancer-causing, some that are apparently random, some in genes we don't even understand yet. The genetic chaos visible in a cancer cell is a direct consequence of losing the checkpoint systems that normally prevent this kind of accumulation. But here's the thing that's important to understand. Of multiple systems. A cell with broken of multiple systems. A cell with broken p53, but no other problems might still not become cancer. It might have a growth disadvantage. It might die from unrepaired DNA damage. It might get caught by the immune system. The body has many layers of defense, and losing one layer doesn't automatically mean disaster. But if that same cell then develops another mutation, maybe in a gene that makes growth hormones, now you have a bigger problem. A cell with defective p53 that also produces too much growth signal. That cell divides more readily than normal cells. It competes better for resources. It doesn't get tired. It doesn't respect the boundaries of the tissue. And if it then develops a third mutation, maybe in a gene that's supposed to trigger cell death when something goes wrong, Now you have a cell that's even more dangerous. 3 mutations, 3 layers of defense removed, 3 different safety systems failing in the same cell. A cell that's not just growing, but growing in defiance of everything that's supposed to control it. This is called the multi-hit model, and it's one of the most important concepts in understanding cancer. Cancer doesn't usually arise from a single mutation. It arises from the accumulation of multiple mutations over time, each one removing another layer of protection, each one making the cell more aggressive and harder to control. Researchers have found that most common cancers require about 4 to 7 key mutations. Not just any mutations, but mutations in genes that directly affect growth control. Mutations in oncogenes that turn on growth signals. Mutations in tumor suppressor genes that turn off growth control. Mutations in genes that control cell death. Mutations that affect how cells respond to their environment. These are the critical hits. The mutations that matter. Most cancer cells also have many other mutations that are just along for the ride, random damage that happened to be copied when the dangerous mutations were copied. But there are typically 4 to 7 key mutations that actually drove the cancer development. This matters because it explains why cancer takes so long to develop. It's not just one unlucky random event. It's a series of events, each one requiring chance, each one increasing the probability of the next. The more years a cell is exposed to mutagenic factors, the more opportunities for mutations to accumulate, the higher the probability that enough key mutations will accumulate in the same cell to cause cancer. The scary part is that this process can take years, decades even. Someone might smoke cigarettes for 30 years without developing lung cancer. The first cigarette doesn't cause cancer. Neither does the 100th or the 1000th. But each cigarette delivers DNA-damaging chemicals into the lungs. Each one might cause a mutation in a lung cell. Most of these mutations get repaired. Most cells with mutations die or are removed by the immune system. But some persist, some accumulate. After decades of exposure, a lung cell somewhere might have accumulated enough mutations that it crosses the line from normal to cancerous. The timing is essentially random, which is why cancer seems to strike unpredictably. The damage is accumulating invisibly for years before anything visible happens. This is called the latency period, and it's one of the reasons cancer is so hard to prevent. You can't see the damage accumulating. You can't point to a specific moment when the cell became cancerous. It's a gradient, not a threshold. A slow transition from normal to abnormal to dangerous. DNA damage is the initiating event in most cancers. And understanding the sources of that damage is crucial because some of them you have some control over. Your cells are being exposed to DNA damaging agents all the time. Some of these are external. UV radiation from the sun, tobacco smoke, certain chemicals in food or water, asbestos fibers, air pollution, radon gas in your basement, pesticides, industrial chemicals, some food additives. These are exogenous sources, meaning they come from outside your body. But a lot of DNA damage comes from inside. Your cells are constantly undergoing metabolic processes that produce reactive molecules, free radicals, that can damage DNA. The normal process of breaking down food produces free radicals as a byproduct. When your immune system fights an infection, it deliberately creates free radicals to kill the invading bacteria. These free radicals are incredibly useful in controlled amounts. They're weaponized defense systems, but they're also dangerous. They can damage DNA indiscriminately. They can damage proteins. They can damage the very protective systems that are trying to neutralize them. Your cells have antioxidant systems to neutralize these free radicals before they cause too much harm. Enzymes like catalase and superoxide dismutase. Molecules like vitamin E and glutathione. These systems are constantly working, but it's an ongoing battle. The damage keeps happening. The cleanup keeps happening. Most of the time, the damage gets repaired faster than it accumulates. Most of the time, the system works. But as you age, the repair systems become less efficient. Enzyme production slows, proteins accumulate damage and work less effectively. The antioxidant defenses become less effective. Free radicals accumulate. DNA damage accumulates. Unrepaired mutations persist in cells. This is why cancer is primarily a disease of aging. The older you are, the more time your cells have had to accumulate mutations, and the more likely it is that one of those cells will have accumulated enough mutations to become cancerous. A newborn baby has essentially zero risk of cancer, even though all the same biological processes are happening. But a 70-year-old has had 7 decades of exposure to DNA-damaging agents, 7 decades of accumulating mutations in cells, 7 decades of cellular selection, where the most aggressive cells gain advantages over normal cells. The risk increases exponentially with age. The repair systems themselves are the first line of defense against this accumulation. When DNA damage is detected, which happens constantly, repair proteins are recruited to the site. These proteins scan the DNA, identify mismatches, identify breaks, identify distortions. They assess whether the damage is repairable. If it is, they perform surgery at the molecular level. They cut out the damaged section, carefully, with precision. They use the undamaged strand as a template to rebuild the missing section. They seal the gaps. They verify that the repair was successful. It's an incredibly complex process, and it works with extraordinary accuracy most of the time. But sometimes it fails. Sometimes the damage is so severe that the repair machinery can't fix it. Sometimes the cell runs out of energy or resources before the repair is complete. Sometimes the repair itself introduces new errors. And critically, sometimes the gene that codes for the repair machinery itself gets damaged. If the gene that codes for a DNA polymerase gets mutated, then the repair system becomes less reliable. If the gene that codes for a proofreading protein gets broken, then errors start creeping through. This is where the cascading failure really becomes dangerous. The system meant to protect against mutations gets broken, so mutations accumulate faster, which makes it more likely that other protective systems will get damaged, which makes mutations accumulate even faster. It's a feedback loop that accelerates the whole process. Some people are born with inherited mutations in their DNA repair genes. They inherit a defective copy of a gene like BRCA1 or BRCA2, which are involved in repairing certain kinds of DNA damage. These people have a much higher lifetime risk of developing cancer because their cells are accumulating mutations faster than normal. The repair systems are fighting an uphill battle from the very beginning. They can still repair most damage, but not all of it. Mutations persist and accumulate. These inherited mutations explain why some families have clusters of cancer. Multiple people in the same family getting cancer at younger ages than average. They inherited the same broken repair gene. But even inherited mutations aren't automatically a death sentence. People with BRCA1 mutations have about a 70% lifetime risk of developing breast cancer. That's much higher than the general population risk, which is around 13%. But it's not 100%. It's not guaranteed. Some people with BRCA1 mutations never develop cancer. Some develop cancer only after living to old age. This suggests that even with an inherited mutation in a repair gene, You still need additional mutations in other genes before cancer actually develops. You still need that multi-hit accumulation. The inherited mutation just makes it more likely that you'll accumulate those additional hits before the repair systems can eliminate the dangerous cells. This is one of the reasons why understanding cancer at the molecular level is so important, because it reveals that cancer is not usually a single catastrophic event. It's not like flipping a switch from normal to cancerous. It's a process, a gradual accumulation of damage, a series of cellular accidents that compound over time. And that process takes years, often decades. Which means there's potentially time to intervene, to catch dangerous cells before they become truly malignant, to remove them before they spread. But only if we understand what to look for, only if we understand how the process works and where it's vulnerable. When a mutation happens in a cell and it doesn't get repaired, that mutation gets copied every time the cell divides. Both daughter cells inherit it. But mutations themselves come in different types, and they have very different effects. A mutation might change a single letter in the 3 billion letter genome. Just one A changed to a G, one tiny change. It might delete a chunk of DNA. A whole section just removed. It might insert extra copies of a gene. Duplication errors. It might reverse the order of a section of DNA. Inversions. It might move a gene from one chromosome to another. Translocations. The physical complexity of these DNA rearrangements is staggering. Some mutations happen in non-coding regions and have no effect at all. These regions don't code for proteins, so changing Hem doesn't change anything critical. Some happen in genes that have redundancy, where you have 2 copies and losing one doesn't matter. Some happen in critical genes and immediately kill the cell or severely impair it so badly that the cell cannot survive. These mutations are usually quickly eliminated because the cell either dies or is removed by the immune system. Some mutations are subtle. They change a single amino acid in a protein, which might slightly alter how that protein works. It might speed it up or slow it down. It might change its shape slightly, or it might have no effect at all. These subtle mutations can be harder to detect and harder to eliminate through natural selection, which means they sometimes accumulate in cells without anyone noticing. The effects are too small to trigger alarms. Other mutations are dramatic. They might completely stop a protein from being made, a premature stop signal in the middle of the gene. They might produce a protein that's toxic or non-functional. They might destroy a crucial function. These mutations are usually quickly eliminated because the cell either dies immediately or is removed by the immune system. The mutations that matter most for cancer development are the ones that affect cell growth and division, and they're often the subtle ones, the ones that don't immediately kill the cell but slowly shift its behavior, the ones that change how the cell interprets growth signals, the ones that break checkpoint mechanisms, the ones that shut down cell death programs. These are the mutations that shift the balance of power in the cell's decision-making processes. A normal cell is constantly receiving signals, some saying divide, some saying stop, some saying grow, some saying prepare to die. The cell weighs these signals. It integrates them. It makes a decision based on the totality of what it's hearing. A cell with a mutation that increases the response to growth signals will divide more readily. Even a weak growth signal will be amplified. A cell with a mutation that decreases the response to stop signals will ignore commands to pause. It won't hear the message clearly, or it won't care. A cell with a mutation in a gene like P53 that prevents it from responding to DNA damage will divide even when it shouldn't. It won't get the alert that something's wrong. Taken one at a time, these mutations might not be catastrophic. A cell that divides slightly more readily than normal might still be controlled. A cell that's less responsive to stop signals might still eventually stop, but in combination, they're dangerous. Each one removes another constraint on cell division. Each one makes it easier for the cell to ignore the signals that keep it in line. And if you stack enough of them together, the cell becomes unbiddable. The process of mutation itself happens all the time. During DNA replication, which happens billions of times per day in your body, mistakes occasionally happen. A copying enzyme might insert the wrong nucleotide, the wrong base pair. Most of these mistakes are caught by the proofreading systems that follow immediately behind the copying machinery. But some slip through, spontaneous mutations they're called. They happen without any external cause. Just random errors in the copying process. Then there are mutations caused by environmental factors. Tobacco smoke contains chemicals called polycyclic aromatic hydrocarbons, PAHs. These chemicals combine directly to DNA and cause mutations. UV radiation from the sun can damage DNA directly, creating thymine dimers, where two thymine bases bond to each other instead of to their partner bases on the other strand. This disrupts the DNA structure and causes replication errors. Certain foods, especially if they're charred or cooked at very high temperatures, can contain compounds that cause DNA damage. Some people inherit genetic variations that make them better or worse at repairing certain types of damage or breaking down potentially harmful chemicals, which shifts their individual risk. But all of it, happens on a background of constant, unavoidable DNA damage that your cells are designed to manage. The crucial point is that mutations are normal. They happen constantly. Your body is designed to live with a baseline level of mutation and repair. The system works. Cells die when they shouldn't be alive anymore. Damaged cells are eliminated. Immune cells patrol the tissue looking for anything that looks wrong. Most potential cancers are caught and eliminated before they ever become a problem. Most cells with multiple dangerous mutations never survive long enough to divide. But every now and then, through a combination of bad luck and accumulated damage, a cell slips through. A cell develops a mutation that removes one layer of protection, then another, and nobody notices. Not for a while. Not until the dangerous clone has divided enough times to create a population of abnormal cells large enough to cause problems. That's when cancer becomes detectable. That's when something that's been happening silently for years suddenly becomes a crisis. Oncogenes are what we call genes that have been mutated or activated in ways that promote cell growth and division, and they're the accelerators in the cancer system. The difference between a normal gene called a proto-oncogene and its dangerous mutated version, an oncogene, is often subtle, deceptively subtle. A proto-oncogene is just a regular gene that helps control normal cell growth. Lots of cells need to divide regularly. Skin cells, blood cells, cells lining your gut. These cells need growth signals to tell them when to divide. They need to know that it's time, that conditions are right, that there's demand for new cells. Proto-oncogenes encode the proteins that create these growth signals or that receive them. They're essential for normal growth and development. Without them, embryos wouldn't develop, wounds wouldn't heal, the body couldn't maintain itself. A growth factor is a molecule that tells a cell to divide. It's produced by nearby cells or by the cell itself. It travels through the tissue, finds a cell with the right receptor, binds to that receptor, and triggers a chain of reactions inside the cell that leads to division. The specificity here is important. A growth factor can only affect a cell that has the right receptor for it. It's like a key that only fits certain locks. A cell lining the gut responds to growth factors that signal that more intestinal cells are needed. A blood-forming cell in the bone marrow responds to growth factors that signal that more blood cells are needed. The same growth factor means nothing to a cell that doesn't have the receptor. This allows the body to control growth in specific tissues without affecting the whole organism. This is normal. This is necessary. Without growth factors, tissues couldn't develop, couldn't repair themselves, couldn't replace old cells. The system depends on it. The system is elegant. But what happens if a mutation causes a gene to make too much growth factor? Instead of the tightly controlled amount, the cell starts pumping out massive quantities. Or what if a mutation causes a receptor to stay activated all the time, even without the growth factor present? The signal is always on, always there. Or what if a mutation causes the signal-sending machinery inside the cell to get stuck in the on position? The message that's supposed to be relayed only when the growth factor arrives is being relayed constantly, always. A cell that's constantly receiving the signal to divide will divide constantly. It doesn't matter what external signals are present. It doesn't matter whether conditions are right for division. It doesn't matter whether there's enough space, enough resources, or enough demand for new cells. It will divide. It cannot stop. This is an oncogene, a proto-oncogene that's been activated inappropriately. It's still the same gene doing the same basic job, but it's doing the job too much or in the wrong context. It's stuck in the on position. An analogy is a car. A normal proto-oncogene is like the gas pedal. It's supposed to make the car go forward when you press it. That's its function. You press, the car goes. You release, the car slows. An oncogene is like a gas pedal that gets stuck, permanently jammed at half throttle. Or one that makes the car accelerate even when you're not pressing it. Or one that makes the engine rev at full power constantly, burning fuel, roaring, regardless of whether anyone is trying to steer or brake or do anything else. The car doesn't stop when you want it to. The car doesn't listen to the brake anymore because the engine is already running at maximum. The steering wheel is useless when you're careening out of control. This is a profound problem. A cell with an active oncogene is a cell in overdrive. Oncogenes can arise in several ways. Point mutations, where a single nucleotide is changed, can alter the gene enough to change how the protein it codes for works. A protein might become hyperactive. A regulatory region might be mutated so that the gene is turned on constantly, instead of on and off as needed. Gene amplification is another mechanism. A cell might accidentally duplicate a proto-oncogene. Instead of 2 copies, it has 4 or 10 or 100. More copies means more protein produced, which amplifies the signal. Chromosomal rearrangements can move a proto-oncogene next to a new regulatory region that turns it on permanently. This happened in a famous example called the Philadelphia chromosome, a translocation event where parts of 2 different chromosomes broke off and swap. Paired places created a fusion between 2 genes, BCR and ABL. This fusion gene produced a protein that was permanently active, constantly sending the signal to divide. This mutation is found in about 95% of chronic myeloid leukemia cases. It was such a specific, identifiable mutation that once researchers understood it, they could develop a drug that specifically targeted this fusion protein, which revolutionized treatment for this leukemia. A single drug that works because it was designed to stop this specific oncogene. The important thing to understand is that an oncogene is only half the problem. A cell with an active oncogene, a cell that's receiving constant growth signals, is dangerous. But it's not necessarily cancer yet. The cell still has to get past the checkpoints. It still has tumor suppressor genes that can tell it to stop. It still has the apoptosis system that can tell it to die. An oncogene on its own, without loss of these other protective systems, often can't cause cancer. The cell with the oncogene might grow faster than normal cells, but it's still constrained by the checkpoints. It's still going to eventually die because cells are supposed to die. It's still going to get eliminated by the immune system if it looks abnormal. But if that same cell, the one with the active oncogene, also develops a mutation in p53, then the checkpoints stop working. If it develops a mutation in apoptosis genes, then it can't be told to die. If it develops mutations in several different tumor suppressor genes, then all the brakes are gone. Now you have a cell that's getting constant growth signals and has no brakes and no capacity to self-destruct. That's cancer. That's the multi-hit model in action. One mutation that promotes growth, one or more mutations that disable growth control, and you have a dangerous cell. Tumor suppressor genes are fundamentally different from oncogenes, and understanding the difference is crucial. Where oncogenes are like stuck accelerators, tumor suppressor genes are supposed to be brakes. They're genes that normally slow down cell division, or trigger cell death when appropriate, or maintain the integrity of the genome. These genes code for proteins that say no to inappropriate cell division. They code for checkpoints. They code for cell death signals. They code for DNA repair enzymes. A normal tumor suppressor gene is intact and functional. It's constantly doing its job, constantly checking on the cell, constantly asking whether conditions are right for division. It's keeping watch. It's vigilant. When a tumor suppressor gene gets mutated, it stops working. The brake is no longer functional. This is qualitatively different from an oncogene mutation. With an oncogene, you have a gene that's on when it shouldn't be. A runaway process, an accelerator stuck. With a tumor suppressor gene mutation, you have a missing brake. There's nothing there to stop the acceleration. There's nothing to slow it down. There's no regulator, just the accelerator, wide open. p53 is the most important tumor suppressor gene. It's mutated in about half of all human cancers, which tells you how critical it is. 50%. 1 out of every 2 cancers has lost functional p53. Sometimes it's mutated in both copies, completely inactivated. Sometimes it's mutated in just one copy, but that one mutation is enough to significantly impair its function, because even partial loss of p53 function creates vulnerability. p53 does multiple jobs. It detects DNA damage through various signaling pathways. It activates repair systems by turning on genes that code for DNA repair proteins. It triggers cell death, apoptosis, if the damage is too severe to repair. It acts as a transcription factor, binding directly to DNA and turning on other genes that are involved in growth control. It's a master regulator. If p53 is broken, all of these functions fail, all of them. A cell with mutated p53 can have severe DNA damage and the cell won't even know. The alarm system is offline. The damage won't get repaired because p53 isn't telling the repair systems to activate. p53 isn't sending the chemical signal. The cell won't die because p53 isn't triggering apoptosis. The self-destruct button is disconnected. The cell will just keep dividing, accumulating more and more mutations with each division because nothing is stopping it, and nothing is checking whether it should be happening. This is why p53 is called the guardian of the genome. It's protecting the integrity of the genome through multiple mechanisms. It's the watchdog. It's the quality control inspector. It's the safety officer. When it's gone, that protection is gone. Li-Fraumeni syndrome is a rare genetic condition where people inherit a mutation in one copy of p53 from birth. They're born with one functional copy and one broken copy in every cell. Having just one functional copy is enough for normal life. Most people have 2 functional copies and never develop cancer. But people with one functional copy have about a 75% risk of developing cancer in their lifetime. 75%. That's not a maybe. That's a high probability. They develop it earlier than the general population. Much earlier. teenagers, 20-somethings, 30-somethings. They often develop multiple independent cancers in different tissues, different organs, sometimes different types of cancer simultaneously. A brain tumor in one place, a breast cancer in another, a sarcoma somewhere else. A person might survive one cancer only to develop a completely different cancer a few years later. This shows how critical p53 is. This shows that losing p53 predisposes cells throughout the entire body to cancerous transformation. Every cell has one broken copy. Every cell is halfway there already. It just needs one more hit, one more mutation to inactivate the remaining functional copy of p53, and boom, that cell has lost a critical layer of protection. The odds of that happening, given decades of exposure to mutagenic factors and billions of cells dividing, become pretty good. It's more likely to become cancerous. It's probably going to become cancerous. And because this is true for every cell in the body, people with Li-Fraumeni syndrome have very high cancer risk and typically develop cancer multiple times over their lifetime. They're fighting against a genome that has a built-in vulnerability. Other tumor suppressor genes are important too, each one protecting against specific types of damage or dysfunction. RB is the retinoblastoma gene, which controls the G1 checkpoint directly. It normally acts like a brake at that critical decision point. When it's mutated, cells can pass the G1 checkpoint without proper verification. They divide when they shouldn't. A PC is involved in limiting cell growth and maintaining normal cell adhesion. When it's mutated, cells start dividing excessively and lose contact with neighboring cells, which is a hallmark of cancer cells. BRCA1 and BRCA2 are involved in DNA repair, specifically in repairing double-strand breaks. These are the most dangerous type of DNA damage because both strands of the DNA helix are broken. When these genes are mutated, the repair system is impaired, and mutations accumulate much faster. A cell with broken BRCA1 or BRCA2 is constantly accumulating damage because it can't fix the most serious type of damage. Loss of any of these genes removes another layer of protection. Each loss makes the cell more dangerous. Loss of multiple tumor suppressor genes, combined with activation of oncogenes, creates a cell that's fundamentally dysregulated. A cell that's lost its ability to be controlled by the normal signals. A cell that's crossed the invisible line from regulated growth to uncontrolled proliferation. That's the moment that cancer begins. Not in years, not in a dramatic moment, but in a single cell that has accumulated enough mutations to become dangerous. A cell that has broken enough of its brakes and hit the accelerator hard enough that it's now a threat to everything around it. But here's what's important to understand about this multi-hit process. It's not just that a cell needs multiple mutations. It's that those mutations need to happen in specific ways, in a specific sequence, and each one needs to be selected for. Not selected by intention, but selected by nature. A cell that develops an oncogene mutation might divide more readily than its neighbors. That gives it a growth advantage. It competes better for resources. It spreads through the tissue more efficiently. Pretty soon, that mutant cell has divided into a clone, a population of identical cells, all descended from the first mutant. They all share that same oncogene. They're all dividing faster than normal, but they're still constrained. They still have tumor suppressors working. They still have checkpoints. They still have the machinery that can trigger cell death when necessary. The clone spreads, but it doesn't become cancer yet. It's just faster than it should be. Abnormal, but not necessarily dangerous. The tissue tolerates this for a while. The clone might expand to thousands or millions of cells. It might occupy a notable section of the tissue, but without additional mutations, without breaching more of the safety systems, it doesn't cross the line into malignancy. A cell biologist looking at these cells under a microscope might notice something wrong. They might be slightly larger than normal. They might have slightly different shapes. They might be more densely packed. But they're not yet uncontrollable. They're not yet spreading beyond their boundaries. They're not yet invading neighboring tissue. They're just a clone of slightly abnormal cells existing in a tissue, held in check by all the remaining safety systems. But now something else happens. One of the cells in that clone, just one, develops a second mutation. Maybe in a tumor suppressor gene, maybe in p53. Now that one cell has both the oncogene and the broken p53. It divides and passes both mutations to its daughters. Those daughter cells have even fewer constraints. They divide faster than the already fast cells around them. They outcompete the mutant cells that don't have the second mutation. They spread through the tissue. And now you have a new clone, descended from the original mutation, but even more dangerous, with 2 hits against normal growth control. The process repeats. A cell in this second clone develops a third mutation, maybe losing another tumor suppressor gene. Now it's even more dangerous. Its clone spreads. One of those cells develops a 4th mutation, and so on. This process takes time, years, decades, because each mutation is rare. Each beneficial mutation in a dangerous direction needs to happen by chance. Each one needs to confer enough growth advantage that it spreads. Each selection takes generations of cell divisions. The first mutation might be a point mutation in KRAS, a proto-oncogene involved in growth signaling. That mutation happens by chance in one lung cell during 30 years of cigarette smoke exposure. That cell divides and creates a clone of maybe 10,000 cells, all with the KRAS mutation. Then, by chance, one of those cells experiences a point mutation in p16, a tumor suppressor that's involved in cell cycle regulation. That cell starts dividing faster than the other mutant cells. Its clone expands. Then another mutation in p53 happens in one of those cells, and another in the APC gene, and another in a cell adhesion gene. Each mutation is rare. Each one requires chance. But the body is full of cells dividing, billions and billions of them every day. Across the entire tissue, across years of exposure to mutagens, the probability that the right mutations will happen in the right sequence eventually approaches certainty. Not inevitability, but high probability. High enough that if you live long enough, a cancer becomes likely. That's why age matters so much. That's why cancer is primarily a disease of aging. Not because old people are weak, but because old cells have had time to accumulate the right mutations in the right sequence. The multi-hit model explains so much about cancer that would otherwise seem like bad luck. It explains why smoking for 30 years sometimes doesn't cause lung cancer, but smoking for 40 years more often does. It explains why some people with genetic predispositions to cancer develop it much younger than the general population. They inherit one mutation, so they're already one hit in. They need one fewer random mutation to reach the threshold of cancer. It explains why cancer is rare in children but common in the elderly. Children haven't had time to accumulate the mutations. Elderly people have. The multi-hit model is also why cancer is so hard to prevent completely. You can't prevent all mutations. You can reduce exposure to carcinogens, which reduces the rate of mutation, which increases the time before cancer develops. You can support your DNA repair systems with antioxidants, with a healthy diet, with sleep and exercise. But you cannot eliminate the baseline rate of spontaneous mutation. You cannot prevent aging. You cannot guarantee that the right sequence of mutations won't happen eventually. The best you can do is shift the odds, reduce the probability, delay the onset. Understanding the multi-hit model also explains why early detection is so important. If a cancer takes 10 mutations to fully develop, then catching it at 3 mutations is much better than catching it at 8. A 3-hit cancer is much less dangerous because it hasn't had time to acquire as many additional mutations. It's still got tumor suppressors working in some of its cells. It's still somewhat constrained by the remaining checkpoints. It's not yet fully adapted to malignancy. Catch it at 8 hits, and the cancer is far more adaptable, more resistant to treatment, more likely to have acquired the ability to spread and the ability to evade the immune system. This is why mammograms, colonoscopies, and other screening tests are valuable even though they're not perfect. They catch cancers earlier in their mutation accumulation when they're still somewhat vulnerable. The multi-hit model also reveals something subtle and important about individual variation in cancer risk. 2 people might be exposed to identical carcinogens for identical periods, but they might have different rates of mutation due to differences in their DNA repair systems. They might have inherited different versions of genes that affect how quickly they metabolize toxins or how effectively they eliminate free radicals. They might have different immune systems, affecting how efficiently they eliminate newly emerged cancerous cells before they become established clones. These individual differences are invisible, but they matter enormously. They're part of why some people smoke their whole lives without getting lung cancer, while others develop it relatively quickly, who inherited a slightly more efficient version of a DNA repair enzyme will accumulate mutations more slowly over their lifetime. A person who inherited a less efficient version will accumulate them faster. They can have identical exposure to carcinogens, identical lifestyle, but one will develop cancer years earlier than the other. This is not fair. This is not justice. This is just biology being indifferent. Understanding this has led to research into biomarkers that might predict individual cancer risk. If you can identify someone who's genetically predisposed to accumulate mutations quickly, you might be able to intervene earlier, screen more intensively, reduce exposure to carcinogens more aggressively. The multi-hit model shifted cancer from being seen as a random catastrophe to being understood as a process, which is much more actionable. Instead of thinking about cancer as something that randomly strikes people, you can think about it as a predictable process that unfolds according to knowable principles. The principles are complex, the variables are numerous, the randomness is real, but the process is not magical. It's not incomprehensible. It follows rules. But the multi-hit model also has a subtle consequence that's worth thinking about. Once a cell has accumulated multiple dangerous mutations, it's not stable. It's unstable in multiple ways. Its genome is unstable because the mutations it's accumulated have often damaged DNA repair systems, making further mutations more likely. Its gene expression is unstable because its regulatory systems are broken. It's constantly trying different combinations of gene activity, and some of those combinations are advantageous and some are lethal. Its evolutionary pressure is extreme because it's dividing constantly, and each division introduces the possibility of new mutations. The cancerous cell is not a fixed entity, it's a population in constant flux. This brings us to clonal evolution. A cancer doesn't arise from one cell with all the necessary mutations perfectly in place. It arises through a series of clonal expansions, each time a new mutation giving a growth advantage that allows one cell's descendants to outcompete everyone else. The cancer is not monolithic. It's a competition between slightly different variants of cancer cells, with the evolutionary pressure being growth and survival under increasingly hostile conditions. The primary tumor, the original cancer at the site where it started, becomes a diverse population. Different cells within the same tumor might have slightly different mutations. A subpopulation might have a mutation in a growth factor receptor that makes them particularly responsive to a certain growth signal. Another subpopulation might have a mutation that makes them resistant to a particular type of immune attack. Another might have acquired the ability to metastasize more efficiently than the others. The tumor is not a uniform entity. It's a heterogeneous population, a collection of different cell types, all with slightly different mutations, all competing under intense selective pressure. Inside the tumor, there's essentially constant evolution happening, not on the timescale of millions of years like evolution in nature, on the timescale of days or weeks. A mutation that gives a slight growth advantage will spread through the clone in a matter of weeks. Think about the math. A cell dividing every day, passing a growth advantage mutation to its two daughters. They divide the next day, creating 4 cells with the mutation, then 8, then 16. In just 10 days, a single mutated cell has become over 1,000 descendants. In 2 weeks, 1,000,000. In 3 weeks, enough to represent a significant fraction of the tumor. The mutation doesn't have to be a huge advantage. Even a 10% faster growth rate will rapidly outcompete the neighboring cells that don't have the mutation. In a matter of weeks, the tumor population shifts toward the new variant, a mutation that makes cells more resistant to the inflammatory environment of the tumor will be selected for quickly. The tumor is constantly under attack by immune cells, T cells, macrophages, natural killer cells. The immune system is trying to stop the tumor. Cells that are killed won't divide. Cells that survive will divide. Cells with mutations that make them less visible to immune cells, or that produce immunosuppressive signals, will survive better. They'll be selected for. A mutation that allows cells to produce new growth factors so they don't have to rely on external signals will be selected for. The tumor becomes a miniature evolutionary system in constant selection. Natural selection operating not at the level of organisms, but at the level of individual cells competing for space and resources inside a tissue. It's Darwinian evolution at cellular scale. This evolutionary process has a consequence that's important for understanding why cancers become so dangerous over time. The cancer that exists in a person's body today is not the same cancer that existed 6 months ago. It's evolved, it's adapted, it's acquired new mutations, it's selected for the variants that are best at surviving in that particular environment. If a particular drug starts killing the cancer cells, the cancer will evolve resistance, not because the cells are trying to, not because they have any awareness of what's happening, but because among billions of cells dividing under intense selective pressure, some will randomly acquire a mutation that makes them less vulnerable to the drug. Those cells will survive and divide while the others die. The cancer population will shift toward the resistant cells. This is why cancers become harder to treat over time. They're not just growing. They're evolving. Melanoma cells in early stages might all have the same mutations. BRAF mutation, loss of p53, whatever the driver mutations are. Uniform, genetically similar, phenotypically similar, all dividing at roughly the same rate, all responding similarly to their environment. But as the melanoma grows and as the immune system attacks it, and as treatment is applied, the cells that survive are the ones with additional mutations conferring advantages. Maybe a mutation that helps them evade the immune system by shutting down MHC expression, making them invisible to T cells. Maybe a mutation that produces PDL1, which sends an inhibitory signal to T cells that would otherwise attack them. Maybe a mutation that amplifies a particular survival pathway like WNT signaling or Notch signaling. The melanoma B, ECOM's a mixed population, different subpopulations with different mutations, different survival strategies, all occupying the same tumor. Then treatment is applied. A drug that targets BRAF, the most common driver mutation. The cells with the original BRAF mutation die. Many of them, most of them. But cells that have also acquired a mutation in a downstream pathway that lets them survive BRAF inhibition will survive. Maybe a mutation in MEK, which is downstream of BRAF. Maybe an amplification of AKT, which is another survival pathway. The population shifts. The cells without resistance die. The cells with resistance expand. The cancer evolves. The tumor composition changes. After a few weeks of treatment, the original BRAF mutant population is mostly gone. The new population is mostly RES-I, istant cells. The cancer has evolved around the drug. This clonal evolution is happening in every cancer, slowly in early tumors where growth is still somewhat constrained, rapidly in advanced cancers where mutation rates are high and selective pressure is intense. The tumor is not a static entity. It's a dynamic population in constant evolutionary flux. Understanding this has changed how researchers think about cancer treatment. Rather than thinking about killing cancer cells, which is impossible to do completely without also killing the patient, researchers think about controlling evolution. Can you apply multiple drugs at the same time so that a cell would need multiple simultaneous mutations to develop resistance? Can you rotate drugs to prevent stable resistant populations from emerging? Can you use immunotherapy in ways that don't create such intense, selective pressure for immune escape? These strategies depend on understanding clonal evolution. The clonal evolution that happens inside a primary tumor also creates the foundation for what happens next, the spread beyond the original site. But before we get to that, we need to understand something else. We need to understand how a small cluster of cancer cells, cells that don't belong in the tissue they're invading, cells that are fundamentally abnormal, manage to grow beyond the natural constraints of the tissue. How do they break free from the normal limits? How do they keep expanding when normal growth control says they should stop? A normal tissue has structure. Cells are held in place by physical contacts with neighboring cells and with the extracellular matrix, the scaffold of proteins surrounding them. This structure is not just mechanical, it's informational. The physical contacts send signals. A cell that's touching other cells gets a signal that there's already enough cells here. Stop dividing. A cell that's not touching the matrix gets a signal that it's in the wrong place. Die. These are called contact inhibition, and it's one of the fundamental constraints on normal growth. A single layer of skin cells grows to a certain thickness and then stops. Not because of external signals, but because the cells in the middle of the layer can't touch the matrix anymore. They receive the signal that they're not supposed to be there. They should die. A layer of cells should be 1 layer thick, 2 at most, not a chaotic heap. But cancer cells have often lost contact inhibition. A mutation in a gene called APC, which is involved in cell adhesion and communicating contact signals, can break contact inhibition. A mutation in E-cadherin, a cell-cell adhesion protein, can do it. A mutation that disrupts the WNT signaling pathway can do it. Multiple different mutations can achieve the same outcome. Cancer cells in culture will pile up on each other, forming layers and layers of cells, stacked haphazardly, because they've lost the signal telling them to stop when they're crowded. They don't care. They divide anyway. They keep piling up. A monolayer becomes a several-layer thick heap. This is called loss of contact inhibition, and it's one of the visible hallmarks of transformation in the lab. In a real tissue, contact inhibition isn't the only constraint on growth. There's physical space. A tumor can only grow so large as the space available in the tissue allows. It's squished by the surrounding normal tissue. The pressure from the tissue pushing inward creates mechanical stress. The tumor is confined. The more it grows, the more pressure it experiences. This physical squeezing actually triggers stress responses in the tumor cells. They experience compression. The stress can inhibit growth. There's also the fact that as the tumor grows, it takes up space that was previously occupied by normal tissue. The normal tissue can only be displaced so far before normal function is disrupted. A tumor in the brain can't grow very large before it starts damaging brain tissue and causing neurological problems. A tumor in the lung can't grow very large before it interferes with breathing. The organ itself imposes limits on growth. There's nutrient limitation. Cells in the center of a cluster are far from the blood vessels that provide oxygen and nutrients. They start to experience hypoxia, low oxygen. Hypoxia triggers stress responses. The cells can't make ATP efficiently without oxygen. Their energy production plummets. Without energy, they can't divide. Under extreme hypoxia, cells start to die. They activate apoptosis. The tumor can grow until it reaches a size where the center becomes hypoxic. Then growth stops. The tumor sits there, unable to expand further, constrained by the diffusion limit. Oxygen can only diffuse about 100 micrometers from a blood vessel. Cells beyond that distance are too far away. They can't get enough oxygen. There's an immune response. The body recognizes that something is wrong. Immune cells infiltrate the tumor. T cells, which directly attack cancer cells. Macrophages, which engulf cancer cells and also produce inflammatory signals. Natural killer cells, which destroy cells that look abnormal. These immune cells attack the cancer cells. The immune pressure limits growth. The tumor is like a foreign invader, and the body's defense systems are trying to eliminate it. A small tumor might be small enough that the immune system can keep it controlled. The immune cells can keep up with the growth rate. The tumor can't outpace them. A slightly larger tumor might overwhelm the immune system's capacity to respond. The tumor grows faster than the immune system can attack it. The immune pressure becomes less effective. But cancer cells have ways of dealing with all of these constraints. Some acquire mutations that shut down immune recognition. They stop displaying the molecular signals that immune cells use to identify them as abnormal. They become invisible. Some acquire mutations that make them resistant to hypoxia. They upregulate genes that let them survive low oxygen. They shift their metabolism to anaerobic processes that don't require as much oxygen. Warburg metabolism, it's called, named after the scientist who first described it. Cancer cells can make energy from glucose even without oxygen, which is less efficient than normal aerobic metabolism. But it works. They can survive where normal cells would die. Some acquire mutations that make them produce anti-inflammatory signals. They recruit immune cells, like macrophages, that don't attack them, but instead help create an environment that supports tumor growth. Some acquire mutations that make them adherent to the matrix in weird ways, allowing them to form three-dimensional structures that are more stable and less dependent on normal tissue architecture. Each of these adaptations removes another constraint. Each one makes the tumor more likely to survive and grow. But none of these adaptations solve the fundamental problem of nutrient limitation. If a tumor grows beyond about 1 millimeter, beyond a certain size, the cells in the center cannot get enough oxygen and nutrients from simple diffusion. They're too far from the blood vessels. The blood vessels that exist are supplying the normal tissue, not the tumor. The tumor cells start to experience hypoxia. They start to die. Growth stops. The tumor sits there, unable to expand further. This is a critical bottleneck. Many cancers never get past this stage. A tumor a few millimeters in size, not enough to be noticed, not enough to cause any problems, constrained by the lack of blood supply. Harmless, from a practical standpoint, because it can't grow any larger. But some cancers cross this barrier. Some cells in the hypoxic center of the tumor develop mutations that cause them to produce angiogenic signals. Vascular endothelial growth factor, VEGF, is the most important. This protein acts as a signal to nearby blood vessels telling them to grow toward the tumor. It's not that blood vessels actively seek out the tumor, it's that the tumor creates a chemical gradient of growth factors that lures the blood vessels in. The concentration of VEGF is high near the tumor and drops off with distance. Blood vessels detect this gradient. They respond, they grow, they branch, they extend new capillaries into the tumor, and as they do, they bring oxygen and nutrients. The tumor cells near the blood vessels have access to resources again. They start to proliferate. The tumor grows. The bottleneck that was constraining expansion suddenly opens. The problem is temporarily solved. The mechanism of how hypoxia triggers VEGF production is elegant and revealing. When tumor cells experience low oxygen, they activate a transcription factor called HIF1, hypoxia-inducible factor 1. This protein binds to the VEGF gene and increases its expression. More VEGF is produced. The more hypoxic the center of the tumor is, the more HIF1 is activated, the more VEGF is produced. It's a negative feedback response, in a sense. The tumor is solving its own oxygen crisis. The deeper the hypoxia, the stronger the signal for blood vessel growth. It's as if the tumor is screaming for help, and the louder the cry, the stronger the response from surrounding tissues. This mechanism is so fundamental that it's conserved across many tissues and organisms. Hypoxia triggers angiogenesis in wound healing. It triggers angiogenesis during fetal development. It triggers angiogenesis during exercise recovery. The system is designed to respond to low oxygen with blood vessel growth. Tumors simply exploit this system. They activate the same signals that would activate angiogenesis in normal circumstances. But this solution creates dependencies. The tumor is now reliant on constant VEGF signaling to maintain its blood supply. It's producing VEGF, sure, but that production has to be constant. If something interferes with VEGF production or VEGF signaling, the blood supply can regress. The tumor can start to die. This is why understanding the mechanism was so important therapeutically. If you can block VEGF, you can cut off the blood supply. If you can block the VEGF receptor on endothelial cells, you can prevent blood vessels from responding to the signal. You're not killing the cancer cells directly. You're starving them. You're withdrawing the resources they need to grow. You're exploiting the dependency that angiogenesis creates. The cancer became addicted to blood vessel growth. Now you're taking away the addiction. But this process creates new problems that the tumor has to solve. The new blood vessels that grow into the tumor are poorly formed. They're not organized like normal blood vessels with proper layers of cells and proper sealing. They leak fluid. They're fragile. They don't provide blood flow efficiently, but they provide enough. They provide just enough blood supply for the tumor to keep growing. The tumor grows larger. More of it becomes hypoxic. The hypoxic cells produce more VEGF. More blood vessels grow in. The tumor expands. This process is called angiogenesis, the formation of new blood vessels, and it's one of the hallmarks of cancer. Without angiogenesis, cancers can't grow beyond a certain size. With angiogenesis, they can grow indefinitely, limited only by the body's capacity to provide blood supply. Understanding angiogenesis led to a whole class of anti-cancer drugs. If you can block VEGF, if you can prevent blood vessels from growing into the tumor, maybe you can starve the cancer. Drugs like bevacizumab, which is an antibody that binds to VEGF and neutralizes it, were designed based on this concept. They don't kill cancer cells directly. They prevent the tumor from getting blood supply. In theory, the tumor should shrivel and die. In practice, it's more complicated. Cancers are adaptive. They find other growth factors that can trigger angiogenesis. If VEGF is blocked, they produce fibroblast growth factor or other signals. They evolve around the blockade. The blood vessels that have already grown into the tumor don't immediately regress. The tumor's relationship to the blood supply is complex, but the principle is sound. Angiogenesis is necessary for cancers to grow large. Blocking it slows growth. In some cases, combining angiogenesis inhibitors with other drugs can improve outcomes. But here's the key insight. Angiogenesis is not something unique to cancer. Blood vessels grow and branch normally throughout life. In wound healing, new blood vessels grow into the wound to supply the healing tissue. The wound creates a temporary chemical gradient of growth factors. The blood vessels respond, grow into the wound, deliver nutrients and oxygen so the tissue can heal. Then, once healing is complete, the growth factors disappear. The angiogenic signal stops. The new blood vessels are pruned back. Angiogenesis ends. In placental development, massive angiogenesis happens to create the blood supply for the developing fetus. The placenta needs nutrients, so it recruits blood vessels. New blood vessels invade the tissue. They provide nutrients for fetal growth. Then, at birth, the placenta is delivered. The angiogenic signal ends. The blood vessel growth stops. In recovery from exercise, new blood vessels grow into muscles to improve their blood supply. The muscle is working hard, creating a signal for more oxygen. Blood vessels grow to meet the demand. This is angiogenesis in action. It's normal. It's essential. Angiogenesis itself is not abnormal. What's abnormal is when it's sustained, when it's directed toward a tumor, when it's not turned off when it should be. Normal angiogenesis is temporary and regulated. Tumor angiogenesis becomes permanent and driven by the cancer cells themselves. The angiogenic signal doesn't stop because the tumor keeps producing VEGF. The blood vessels don't get pruned back because the tumor keeps signaling for growth. The angiogenesis becomes pathological. It becomes a system supporting tumor growth indefinitely. The tumor actively recruits blood vessels. It produces VEGF and other growth factors. Fibroblast growth factor, FGF. Platelet-derived growth factor, PDF. Angiopoietin. These factors have slightly different functions, but they all work toward the same goal. Blood vessel growth. FGF, for instance, works through a different receptor than VEGF. It might be produced by different cells in the tumor, but it reinforces the same message. Grow. It also produces compounds that increase vascular permeability, making it easier for blood vessels to leak fluid into the tumor. This fluid leakage actually helps tumor growth because it brings in nutrients and growth factors. It brings in plasma proteins that can be used by the tumor. It recruits immune cells that normally promote angiogenesis. Macrophages, for instance, produce their own angiogenic factors. Tumor-associated macrophages, they're called, and they often shift toward a phenotype that supports tumor growth rather than attacking it. The tumor essentially hijacks the normal inflammatory response, turning immune recruitment into a process that supports tumor growth. It's a masterclass in biological manipulation. It creates a microenvironment that basically screams to nearby blood vessels. Grow here, expand here, bring more nutrients here. And the blood vessels respond. They grow, they expand, they proliferate. They extend new capillaries into the tumor tissue. Following the chemical gradient of growth factors, they provide the blood supply that allows the tumor to expand beyond the diffusion limit. This bidirectional relationship, the tumor signaling to blood vessels, the blood vessels responding, the tumor benefiting, creates a critical stage in cancer development. Once angiogenesis starts, the tumor can grow much larger. Once it's feeding itself through its own blood supply, it's no longer constrained by diffusion. It's no longer limited to a few millimeters. It can grow centimeters. It can become a palpable mass that can be felt. It can become visible on imaging. It can become genuinely dangerous. It can become something that has real consequences for the organism it's growing in. It's crossed a threshold from microscopic to macroscopic, from invisible to detectable, from contained to threatening. But growing a tumor is only part of the problem the cancer has to solve. Or maybe it's only part of the problem from the cancer's perspective. From your perspective as the organism hosting this growing population of malignant cells, it's the full problem. The tumor is already dangerous. It's consuming resources. It's displacing normal tissue. It's producing signals that induce inflammation. In many cases, early tumors are caught and removed surgically before they cause serious damage. Mammography catches breast cancers. Colonoscopy catches colon cancers. Dermatology catches melanomas. Many cancers are stopped before they ever progress to the next stage. But some escape. Some tumors don't stay localized. Some develop the ability to invade beyond their original boundaries. They break free from the tissue that contains them. They enter the bloodstream or the lymphatic system. And once they're in circulation, once they're traveling through the body, they become something far more dangerous. Metastatic cancer. Cancer that's spread. And this is where the majority of cancer deaths come from. Not from the primary tumor, the original cancer at the site where it started, but from metastases, tumors that have seeded themselves in distant organs. Invasion requires a particular set of changes in the cancer cells. Normal cells are held in place by physical and chemical attachments to their neighbors and to the extracellular matrix. These connections are mediated by proteins called adhesion molecules. E-cadherin is one of the most important. It's a cell-cell adhesion protein that holds epithelial cells, cells in the outer layers of tissues, tightly together. Cells that are strongly adherent to each other form a cohesive tissue. They create a boundary. They create a structure that stays in place. Epithelial tissue, tissue that lines the surfaces of organs and bodies, is built on the principle of tight cell-cell adhesion. That's what makes it epithelial. That's what keeps the cells organized into functional sheets that can absorb, secrete, and protect. Without adhesion, epithelium is just a collection of individual cells. It loses its function. Many cancers, especially early cancers, retain these adhesion molecules. They're still stuck together. They're still organized. They're not yet invasive, but some cells in the tumor develop mutations that disrupt E-cadherin or other adhesion molecules. The mutations can directly damage the protein, making it non-functional, or they can affect the genes that control E-cadherin expression, turning down production. Or they can activate transcriptional factors that suppress E-cadherin expression. The mechanisms vary, but the outcome is the same. Without E-cadherin, the cells lose contact inhibition in a physical sense. They're no longer glued to their neighbors. They can move relative to each other. They become plastic, flexible. They can slip past each other and explore new territory. What was an organized epithelial tissue becomes a mob of individual cells, each pushing against the others, each looking for escape routes, each capable of migrating independently. This loss of adhesion is often coupled with another set of changes called the epithelial mesenchymal transition, or EMT. This is a process where epithelial cells, which are normally fixed in place, organized in layers, and relatively immobile, transition toward a mesenchymal phenotype, which is mobile, invasive, and capable of migration. The transition involves a coordinated change in gene expression. Cells that undergo EMT lose epithelial markers like E-cadherin and gain mesenchymal markers like vimentin and N-cadherin. They change their shape, becoming more elongated, more flexible. They produce enzymes called matrix metalloproteinases, MMPs, which can degrade the extracellular matrix, the protein scaffold that holds tissues together. MMP2, MMP7, MMP9, and others attack different components of the matrix. They cleave collagen, they degrade basement membrane proteins, They create gaps in the matrix structure. Without a solid matrix, tissues lose their structure. The cells can move through the degraded tissue more easily. They can invade. The process is also reversible. Cells that have undergone EMT can undergo the reverse process, mesenchymal epithelial transition, or MET, where they regain adhesion molecules, lose migratory capability, and settle into a new location. EMT is not a cancer-specific process. During embryonic development, cells regularly undergo EMT to allow migration and tissue remodeling. Cells in the neural crest undergo EMT to spread through the developing embryo. Cells in the mesoderm undergo EMT to migrate to their proper locations. Hearts, lungs, and brains are built using EMT. The process is normal, essential. What's abnormal is when cancer cells activate EMT as a way to escape from the tissue that contains them. What's abnormal is when cells that should stay put start running. The transition is often triggered by signals from the tumor microenvironment. Hypoxia, the low oxygen at the center of larger tumors, can trigger EMT. Inflammatory signals from immune cells infiltrating the tumor can trigger EMT. Growth factors produced by fibroblasts and other stromal cells in the tumor microenvironment can trigger EMT. The cancer cells are essentially being told to move, to invade, to escape by signals from the environment around them. Once a cancer cell undergoes EMT, it becomes capable of leaving its original location. It can move through tissue, degrading the matrix as it goes. It can invade neighboring tissues. It can breach blood vessels and enter the circulation. This process is called intravasation, the entry of tumor cells into blood vessels. Once in the bloodstream, the tumor cells are traveling through the body. Most of them will die. The bloodstream is a hostile environment for cancer cells. The cells are exposed to immune cells that can attack them. Natural killer cells, in particular, are on patrol in the bloodstream. They recognize abnormal cells and trigger their apoptosis. T cells flow through the blood too, looking for cells displaying antigen. They're exposed to fluid shear stress as blood flows past them at significant velocity. The mechanical stress of flowing blood can damage cells. Red blood cells, white blood cells, platelets are all colliding with the circulating tumor cells. They're exposed to reactive oxygen species produced by normal metabolism. Free radicals in the bloodstream damage cell membranes. They induce oxidative stress that triggers apoptosis. Most circulating tumor cells undergo apoptosis, programmed cell death. They can't survive the journey. The lifespan of a circulating tumor cell is often measured in minutes to hours. It's one of the harshest environments in the body from the perspective of a cancer cell. But some survive. Some cells have mutations that make them more resistant to stress. Mutations in survival pathways that let them endure oxidative stress and mechanical stress. Some have mutations that allow them to evade immune attack, mutations that reduce antigen presentation or produce immunosuppressive signals. Some develop the ability to form clumps with platelets or other cells, creating aggregates that are harder for immune cells to attack. A cell hiding inside a platelet clump is harder to kill than a cell by itself. These are the cells that survive circulation. They're selected for in the truest evolutionary sense. The cells that are most likely to survive the hostile environment of the bloodstream are the ones most likely to reach a distant organ and establish a metastatic colony. The ones with the strongest survival pathways. The ones most capable of resisting stress. The ones most adapted to life in a fundamentally dangerous environment. When a circulating tumor cell reaches a distant organ, a new set of challenges emerges. The cell has to survive in an unfamiliar tissue, with an unfamiliar chemical environment, with unfamiliar neighbor cells, with an immune system that's ready to attack it. Most cells that reach a distant organ either die immediately or enter a dormant state, just sitting there, not dividing, not producing any signals that would alert the immune system to their presence. They're like sleeper cells in a spy movie, waiting for conditions to favor their activation. But some cells begin to divide. They establish themselves in the new tissue. They begin to create a secondary tumor. For this to happen, the cells have to adapt to the new tissue. They have to produce the right growth factors to stimulate angiogenesis in the new location. They have to establish connections with local cells. They have to modify their behavior in ways that make them compatible with the new environment. This adaptation is not guaranteed. Many cells that seem to establish themselves remain small, microscopic, dormant. They don't grow. They don't progress to macrometastases, tumors large enough to cause symptoms or be detected. The reason is often immune control. The immune system keeps the dormant tumor under control. The cancer cells are present, but they're not expanding because the immune system is holding them in check. The equilibrium can last for years. A patient can be in remission, with no signs of cancer, while dormant tumor cells are still present in distant tissues, held in control by the immune system. But the equilibrium can break. If the immune system becomes compromised, if surveillance slips, if the tumor cells acquire new mutations that make them less visible to immune attack, the dormant cells can begin to divide. They can expand. They can become clinically apparent. This is why immunosuppression, whether from age, from disease, or from medical treatment, can increase cancer recurrence. The immune system is not just fighting active tumors. It's continuously surveilling for small populations of cancer cells that might otherwise establish themselves. Once a metastasis does establish, the process accelerates. The cells in the metastatic tumor face the same selective pressures as the primary tumor, the same competition for resources, the same need for angiogenesis, the same vulnerability to immune attack, and the same opportunity for clonal evolution. The metastatic tumor often progresses faster than the primary tumor because the cells seeding it are already far more adapted. They've survived the journey through the bloodstream. They've established themselves in a new tissue. They've acquired mutations that make them more aggressive, more resistant to stress, more capable of survival in hostile environments. The metastatic population is like the second generation of a species introduced to a new environment. It's had time to adapt. It's more likely to thrive. This is why treating cancer becomes so much harder once it's metastatic. A primary tumor is localized. It's in one place. If you can reach it, if you can cut it out surgically or kill it with radiation or chemotherapy, you've potentially eliminated the entire cancer. But a metastatic tumor has seeded multiple sites. There might be one metastasis or dozens or hundreds, depending on the cancer type and how advanced the disease is. You can't surgically remove them all. You can't irradiate them all. Treatment has to be systemic, affecting the whole body, not just local sites. And systemic treatments are harder to tolerate. They affect normal cells as well as cancer cells. The margin between therapeutic benefit and toxicity is narrower. The spread of cancer through the body is not random. Different cancers tend to metastasize to particular organs. Breast cancer often spreads to the lungs, bone, liver, and brain. Colon cancer often spreads to the liver. Melanoma often spreads to the lungs and brain. This pattern is not accidental. It reflects the biology of the cancer cells and the susceptibility of different tissues. A cancer cell that's already adapted to the tissue where it originated may be better adapted to tissues with similar properties. A cancer that evolved in the lungs, a tissue with particular blood flow patterns and immune composition, may be more likely to survive in another lung tissue than in brain tissue, or it may be that the first place a circulating tumor cell reaches is the lungs, simply because all blood from the lower body drains through the right side of the heart, which pumps blood directly to the lungs. The first filter a cancer cell encounters after entering the bloodstream from most tissues is the lung. So lung metastases are common for cancers from many origins. The pattern of metastasis reflects both the biology of the cancer and the anatomy of circulation. Understanding invasion and metastasis has led to new research strategies aimed at preventing spread. If you can prevent EMT, if you can interfere with the signals that trigger cells to become invasive, maybe you can stop cancers before they escape the tissue that contains them. If you can prevent intravasation, if you can stabilize blood vessels so tumor cells can't break through, maybe you can reduce the number of circulating tumor cells. If you can enhance immune surveillance, keeping dormant metastases under control, maybe you can prevent recurrence. These are not yet proven strategies. Cancer's ability to evolve and adapt makes it difficult to block any single pathway. But the logic is sound. The earlier you can intervene in the metastatic process, the better the chances of controlling the cancer before it becomes truly systemic. While the tumor is establishing itself and spreading, it faces a constant threat from the very system designed to protect the organism from invaders. The immune system. Cancer cells are fundamentally abnormal. Their DNA is damaged. Their genes are mutated. Their behavior is aberrant. They produce proteins that normal cells don't produce. In theory, the immune system should recognize them as threats and eliminate them immediately. And in fact, this is probably happening constantly. Most cells that acquire a single oncogenic mutation are probably recognized and destroyed before they can divide into a clone. Most cells that acquire a few dangerous mutations are probably detected by immune cells before they expand further. The immune system is continuously surveilling tissues, looking for cells that behave abnormally. T cells, white blood cells that directly attack infected or abnormal cells, are patrolling tissues constantly. They're looking for cells that display unusual proteins on their surface. Cancer cells, by definition, are cells with unusual proteins. They've got mutated versions of normal proteins. They've got proteins from oncogenes that are overexpressed. They've got neoantigens, totally novel proteins created by frameshift mutations or fusion events. These are obvious targets for the immune system, but cancers that grow large enough to be clinically apparent have already evaded immune destruction at early stages. They've acquired mutations that allow them to hide from or resist immune attack. One strategy is to reduce the presentation of tumor antigens. Cancer cells can mutate genes involved in antigen presentation, the process by which cells display abnormal proteins on their surface so that immune cells can recognize them. A mutation that disrupts the MHC system, the major histocompatibility complex that displays antigens, makes the cancer cell invisible to T cells. The T cells can't recognize a threat if they can't see it. Without antigen presentation, even a highly abnormal cell looks normal to the immune system. Another strategy is to produce immunosuppressive signals. Cancer cells can produce cytokines, signaling proteins, that recruit immune cells with anti-inflammatory functions, macrophages that suppress inflammation rather than promote it, T regulatory cells that dampen immune responses, myeloid-derived suppressor cells that actively inhibit T cell function. These cells are attracted to the tumor and end up supporting tumor growth rather than attacking it. The tumor essentially weaponizes the immune system against itself. It hijacks the recruitment mechanisms that normally bring immune cells to sites of injury or infection and redirects them towards supporting the tumor. It's a masterpiece of biological manipulation. The tumor doesn't just hide from immunity. It doesn't just resist being killed. It actively recruits immune cells that suppress other immune cells. It turns the immune system's own workers against immunity. Some cancers produce PD-L1, a protein that binds to receptors on T cells and sends them a signal to stop attacking. It's like the cancer cells are sending an inhibitory signal to immune cells, telling them to stand down. The T cells see this signal through their PD-1 receptor and reduce their attack. They become exhausted, incapable of killing the cancer cells they're in contact with. They might be in direct physical contact with cancer cells, the closest they could be, and yet unable to mount an effective attack because the inhibitory signal is overwhelming the activating signal. Some cancers produce TGF-beta, a growth factor that inhibits the immune system. Some cancers produce TGF-beta, a growth factor that has broad immunosuppressive effects. It downregulates the function of natural killer cells, reduces T-cell infiltration, and promotes the differentiation of T regulatory cells. The tumor is essentially creating an immunosuppressive microenvironment around itself. Everything it produces tends to suppress immune function. The immune system tries to attack the tumor, but the tumor is constantly sending signals that dampen the attack. The immune cells that do infiltrate the tumor are often skewed toward anti-inflammatory phenotypes, macrophages that support tissue remodeling and angiogenesis, rather than attacking the cancer cells. B cells that produce antibodies that don't effectively neutralize the cancer cells. The tumor has not eliminated the immune response. It's redirected it. It's co-opted it. It's turned immunity into immunity that supports cancer growth. This is one reason why cancer is so difficult to treat with the body's own immune system. The tumor is not just hiding from immunity. It's actively recruiting immunity that helps it grow. The cancer hasn't just evolved to avoid being detected. It's evolved to avoid being attacked even when detected. Even when immune cells are physically present in the tumor. Even when T cells are in direct contact with cancer cells. The cancer has multiple mechanisms to prevent being killed. Some cancer cells have mutations that prevent apoptosis. They've lost p53 or other genes involved in programmed cell death. Even if immune cells trigger the apoptotic pathway, the signal has no effect. The cancer cell can't die because it's lost the ability to commit suicide. Some cancers produce anti-apoptotic proteins like BCL2 that actively block the apoptotic pathway. Even if immune cells do everything right, do everything they're supposed to do to trigger cancer cell death, the cancer cell can simply refuse to die. The signal arrives but bounces off a wall of anti-apoptotic proteins. Understanding immune evasion has revolutionized cancer treatment. If the problem is that cancer cells are hiding from the immune system by reducing antigen presentation, maybe you can upregulate antigen presentation. If the problem is that cancer cells are sending inhibitory signals to immune cells, maybe you can block those signals. Checkpoint inhibitors, like anti-PD-1 antibodies, work by blocking the interaction between PD-L1 on cancer cells and PD-1 on T cells. Without that interaction, without the inhibitory signal, the T cells are free to attack the cancer. They become activated again. They kill the cancer cells. This approach has proven remarkably effective for some cancers, melanoma especially, where the immune system can be reactivated to attack the tumor. Display so that immune cells that were attacking the old antigens no longer recognize them. They can produce different immunosuppressive cytokines if PD-L1 is being blocked. They can develop mutations that make them less susceptible to T-cell killing mechanisms. The evolutionary pressure is intense. The immune system is attacking. The cancer cells that survive the attack are the ones with new mutations conferring resistance. They expand, they become the dominant population. The cancer has evolved around the immune attack, just as it evolved around other selective pressures. This is the constant theme underlying cancer's lethality. It's not that cancer cells are uniquely powerful or invulnerable. It's that they're rapidly evolving. They're acquiring mutations at a rate far higher than normal cells. They're under intense selective pressure. The cells that survive are the ones most adapted to their current environment. If the environment changes, if a new treatment is applied, if the immune system becomes activated, the cancer evolves. It finds new solutions. It develops new mutations. It escapes the threat. The human body is continuously trying to kill the cancer. The cancer is continuously evolving to survive. The tumor is not just cancer cells. It's an ecosystem. The primary tumor that's growing in the body contains not just malignant epithelial cells, but also fibroblasts, immune cells, blood vessel cells, and a complex extracellular matrix. These non-cancer components make up a substantial fraction of the tumor, sometimes the majority. A tumor sample that looks like it's entirely cancer cells under the microscope might actually be only 50% cancer cells and 50% stromal cells, the supporting tissue surrounding and infiltrating the cancer. In some tumors, the stromal fraction is even higher, leaving cancer cells as a minority in their own tumor. The fibroblasts in particular play crucial roles. They're recruited to the tumor by signals produced by cancer cells. The cancer cells secrete growth factors like fibroblast growth factor and transforming growth factor beta. These signals say to nearby fibroblasts, "Come here, help us grow." And the fibroblasts respond. They migrate toward the signal. They migrate into the tumor. Once there, they produce growth factors, cytokines, and extracellular matrix components that support tumor growth. They produce HGF, hepatocyte growth factor, which promotes cancer cell growth and invasion. They produce VEGF, vascular endothelial growth factor, which promotes angiogenesis. They produce IL-6, interleukin-6, which promotes inflammatory responses that support tumor growth. The cancer cells tell the fibroblasts what to do, and the fibroblasts comply, building the infrastructure that the cancer needs. Some of these fibroblasts are recruited from distant locations. The cancer sends out signals that recruit mesenchymal stem cells from the bone marrow, circulating progenitor cells with the capacity to differentiate into fibroblasts. These cells migrate to the tumor and differentiate. They become tumor-associated fibroblasts, CAFs. They're not part of the original tumor. They're recruited from the normal tissues and reprogrammed. The cancer has essentially stolen cells from the body's own repair mechanisms and turned them towards supporting tumor growth. These cells would normally help heal wounds. Instead, they're helping grow a tumor. The immune cells infiltrating the tumor are another key ecosystem component. Macrophages, T cells, natural killer cells, dendritic cells, they're all there. They're trying to attack the tumor, but many of them end up supporting tumor growth instead. The macrophages can be polarized toward anti-inflammatory phenotypes that support angiogenesis and tissue remodeling. The T cells can become exhausted, unable to sustain an attack. The dendritic cells can fail to activate anti-tumor immunity effectively. The cancer has not eliminated these cells. It's corrupted them. It's turned them into unwilling accomplices. The extracellular matrix within the tumor is different from the matrix in normal tissue. It's often denser, more cross-linked, more rigid. Collagen fibers, which form the backbone of most matrices, are heavily cross-linked through enzymatic processes. Lysyl oxidase, an enzyme produced by fibroblasts and cancer cells, oxidizes lysine residues in collagen. This oxidation allows the lysines to form covalent crosslinks with other collagen molecules. The result is a much stiffer, more rigid matrix. This might seem like it would prevent invasion, but it actually does the opposite. The rigid matrix can increase mechanical signaling to cancer cells through integrins, proteins that link cells to the matrix. Mechanical stress from a stiff matrix can activate growth and survival pathways in cancer cells. It tells them to divide. It tells them to survive. The matrix is also remodeled in ways that facilitate invasion. The fibroblasts and MMPs degrade specific areas of the matrix, creating corridors for cancer cells to invade through. The matrix is degraded strategically, not uniformly. There are areas where the matrix is dense, providing structural support to the tumor. There are areas where the matrix is thin, providing escape routes. The tumor essentially carves pathways through its own matrix. There are also areas where specific matrix proteins are overexpressed, proteins that support cancer cell survival and migration. Fibronectin is overexpressed in many tumors, and cancer cells have receptors for fibronectin that signal for survival. Osteopontin is overexpressed in some tumors, promoting cell survival and preventing apoptosis. These are not random changes, they're coorder, innated remodeling. The matrix is not inert. It's not just a scaffold. It's an active participant in the tumor ecosystem. It's signaling to cancer cells. It's being remodeled by cancer cells and fibroblasts. It's a living, dynamic structure that's participating in tumor growth. Immune cells, fibroblasts, cancer cells, blood vessels, and extracellular matrix all interact. They signal to each other. They influence each other's behavior. The tumor is a community. It's a system where multiple cell types, multiple biochemical signals, multiple mechanical forces all interact to create an environment that supports cancer growth. Understanding the tumor as an ecosystem has led to new therapeutic approaches. If the problem is that fibroblasts are supporting tumor growth, maybe you can target fibroblasts, block the signals that recruit them, or reprogram them. If the problem is that macrophages are supporting angiogenesis, maybe you can change their phenotype from pro-tumor to anti-tumor. If the problem is that the matrix is supporting growth, maybe you can modulate the matrix, making it less supportive of cancer cells. These strategies are still largely experimental. Cancer's heterogeneity and complexity make it difficult to target all the ecosystem components simultaneously. But the principle is sound. The tumor depends on support from its ecosystem. Eliminate that support, and you potentially eliminate the cancer's growth. The complexity of cancer biology extends far beyond the mechanisms we've described. Every statement we've made is true in some cancers and not in others. Some cancers don't need angiogenesis because they're located near existing blood vessels. Some cancers don't suppress the immune system very much because they're already genetically distinct enough that the immune system can recognize them. Some cancers are highly dependent on particular growth factors and can be killed by blocking those growth factors. Others are genetically promiscuous, equally capable of using multiple different growth pathways to survive. If you block one pathway, they activate another. The heterogeneity of cancer is one of its defining features. There are over 200 different types of cancer, with subtypes numbering into the thousands. Cancers of epithelial tissues like skin, lung, colon, breast. Cancers of immune cells like leukemia and lymphoma. Cancers of connective tissue like sarcomas. Cancers of endothelial cells like hemangiosarcoma. Cancers of germ cells. Cancers of neural tissue. Each type has unique biology, Each type evolved from a different normal cell type in a different tissue with a different set of normal growth constraints. So they break those constraints in different ways. Even within a single cancer type, there's enormous variation. Two patients with the same histological diagnosis of lung adenocarcinoma might have completely different sets of mutations driving their cancers. One might have a KRAS mutation as the primary driver. The other might have an EGFR mutation. A third might have an ALK translocation. A fourth might have a BRAF mutation. These are all different mutations, activating different pathways, requiring different treatments. One patient might have high tumor burden in the lungs with minimal immune infiltration, a tumor that's growing rapidly but not triggering much immune response. The other might have lower tumor burden but extensive immune infiltration, a tumor that's smaller but highly visible to the immune system. The treatments that work for one might not work for the other. The same chemotherapy drug might shrink one tumor and have no effect on another. The same targeted therapy might work brilliantly for one mutation and be completely ineffective for another. Cancer is not a single disease. It's a collection of diseases, each defined by a particular set of mutations, each with a particular biology, each requiring a particular set of treatments. The heterogeneity extends even within individual tumors. Different regions of a single tumor can have different mutations. Different cancer cell clones within the same tumor can be competing with each other. The tumor is not monolithic. It's a collection of diverse cell populations. This heterogeneity is actually an advantage for the cancer. If a treatment targets one mutation or one pathway, there are probably other cell populations in the tumor that don't have that mutation or don't depend on that pathway. Those populations will survive treatment and expand. The tumor evolves around the treatment. Resistance emerges. This is why combination therapy makes sense. If you use multiple drugs targeting different pathways, the cancer would need multiple simultaneous mutations to develop resistance to all of them. If you use drugs sequentially, rotating through different treatments, you might slow the emergence of resistance by preventing stable resistant populations from establishing. If you use drugs that target different aspects of tumor biology, you might be able to control growth, even though complete elimination is impossible. The goal is not necessarily to cure cancer completely, but to convert it from an acute fatal disease to a chronic, manageable disease. Keep the cancer under control. Manage its growth. Allow the patient to live with the cancer rather than die from it. This is already being accomplished. D for some cancers. Chronic myeloid leukemia is now almost universally manageable with targeted therapy because the BCR-ABL fusion protein that drives the cancer is a single specific target. Some breast cancers with HER2 overexpression are now manageable because HER2 is a single specific target. But for many cancers, where the biology is more complex and the genetics are more heterogeneous, this level of control is still not achievable. The cancer develops resistance to one drug, then another, then another. The patient's condition deteriorates. The disease becomes fatal. The battle between cancer and the body is ongoing, and it's not one that the body always wins. Understanding cancer at the cellular level has revealed just how complex the disease is. It's not a problem of one bad gene. It's a problem of multiple genes working together, of ecosystems supporting growth, of constant evolution and adaptation. It's a problem of cells breaking free from the constraints that normally contain them, invading other tissues, surviving in hostile environments, evading the immune system. Every cancer is a collection of unique mutations, finding unique solutions to the problem of survival in a particular body at a particular time. Understanding these mechanisms has led to better treatments, to better survival rates, to the conversion of some previously fatal cancers into manageable chronic diseases. But it has also revealed the profound complexity of the problem. Cancer is not going to be solved by a single magic bullet. It's going to require ongoing adaptation, ongoing research, ongoing refinement of treatments as cancers evolve and develop new strategies for survival. The biology is elegant in its way, cells finding solutions to survival problems, mutations conferring growth advantages, natural selection operating at cellular scales. But that elegance is cold comfort to the millions of people whose cells are breaking free, whose tumors are growing, whose metastases are spreading. The human cost of cancer is measured in years lost, in suffering, in families disrupted. Understanding the mechanisms is crucial, but understanding them is not the same as having solved the problem. Cancer remains one of the leading causes of death in the world. It remains a disease whose ultimate solutions are still being developed, still being refined, still waiting somewhere in future research and future medicines yet to be discovered.